So we're going to finish up our last section on evolution today by looking at embryology and some biochemical data that we can use to determine evolutionary relationships between organisms. So how can we examine all the structures organisms have over their lives? The first thing that we'd actually look at is embryology. And embryology is the study of how multicellular organisms grow and develop. Just as a little reminder, growth, when we speak of growth in biology, we're talking about cell division, the increase in numbers of cells. And then when we talk about development, we're talking about differentiation, usually due to gene regulation. Often development will also indicate some form of complexity that's occurring in the organism. When you're looking at this picture on the right hand side, there's a couple things that we see here. Obviously several different organisms and different stages within their development. On the x-axis, you see this word ontology, and ontology literally means the development of an individual. And here on the y-axis, we're actually comparing the phylogeny, and phylogeny means the evolutionary branching. Phylogeny refers to the evolutionary branching or the descent between those organisms and relationships between those organisms. So here you can already see some similarities between the fertilized eggs of humans, monkeys, pigs, all the way to the salamander. And then later on when the cells begin to cleave or separate themselves, and even in their body segments, and the fact that we have this limb bud stage. All of these are kind of indicators or patterns, and that's what we're actually using here. We're going to use patterns to show relationships among the organisms. These relationships are going to be based on the appearance or disappearance of specific traits. Examples that we see in this image right here are the pharyngeal slits. You can kind of see some of them along the sides of the organisms, little slits, almost like an indicator of a gill slit. That's the pharyngeal slit. These are similar to gills. And in humans, they actually become part of the nerve covering. So there is a use, but it's not so not the same as how, for example, a salamander might use it. Here, when you're looking at the late cleavage in the organisms, you'll notice that there is a pattern. The pattern that we see is called a radial pattern, and this does indicate, shows relationships among the organism. But just like any of the traits that we've seen so far, whether that be the embryology, the vestigial structures, the homologous structures, all of these structures really come from the DNA basis of those organisms. So what resource can provide the least biased form of evidence? That's going to be our biochemical evidence, which is going to be our DNA, RNA, and amino acid sequences. These are the least biased. You're not just looking at different images or different structures, but you're getting hardcore data here. In biochemical similarities, we're going to compare those sequences. And what you're going to see is the more similarities will lead to more likely connection or relationship amongst those organisms. A lot of times you'll see sequences of DNA, and if you look at this first sequence, top to bottom, we're comparing two different organisms. You have C, A, G, C, A, G, 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 A, and then here you see a G, and we keep going until you get right here, where the star is actually missing. And this is when we first have a difference in sequence. Many organisms, many animals across the animal kingdom have very similar sequences in their DNA, but the few differences will account for some pretty large differences in the organism itself. There's this idea of conserved domains. Conserved domain really means that we have little change, it could be no change, in the genes that organisms have. And this could be due to the fact that the gene is so necessary for life or survival that mutations may not survive. So between different organisms, different animals, different bacteria, sometimes there's some big similarities. Sometimes there's only one big difference, or just a very few differences compared to the thousands of nucleotides or nitrogen bases that you would have in their DNA. An example of a gene that is like this would be DNA polymerase. Okay, the enzyme that actually assembles our DNA molecule. When you're actually looking at the DNA polymerase of different organisms, you'll see there's lots of similarities between them. And science believes, or science thinks, that this is because DNA polymerase is so important. And if there were mutations in DNA polymerase, well, that organism won't survive. And if it can't survive, it won't be there to pass on its DNA. In this next image, or last image really, this is a example of a phylogenetic tree. Some of you have already seen these in some of the work that we've done before, and these show evolutionary relationships between organisms. The specific example says on the left-hand side the number of amino acid differences compared to human cytochrome C. So we're looking at the amino acid sequence here, not the DNA or RNA, although those we know have a role in that. It tells us over here that cytochrome C is a small protein that plays an important role in the electron transport chain within the mitochondria of cells. So basically meaning this is used in respiration. And they're showing us here that the number of amino acids differences between the humans and the monkeys and the pigs and the ducks. And you can see kind of where those differences lie. And the differences are on this vertical axis. So the yeast, if we're starting here, is significantly different from the human over here. Whereas we already know of our huge relationship or connection to monkeys, primates, that there's very little difference between the two. What you want to go ahead and make sure you do is start on, if you already haven't already, your study guide for this unit. I would definitely take time to look back over some of the review questions, maybe even go back, open up the links for the notes, and click on some of the extra resources that we have available for you. And make sure you begin studying for your test. Don't forget to finish up your note cards and answer the rest of the questions on Power Tool. on lecture tools.